Let's continue with our examination of vision. Let's talk about what exactly happens. I've mentioned several times that the cornea does most of the focusing of light and the lens does the rest. Let's look and see how that happens. So light waves have to be refracted or bent to focus on the retina. If you've ever taken a physics class, you know something about this. The refraction of light is the bending of light. Different media have different indices of refraction. So water and air, for example. That's why if you have a stick and it's partway below the water and above the water, it looks like, it goes a different place below than above. That's because media bend light. And what has to happen is, think about it, you're looking at a tree. You can't fit the entire tree onto your retina. So what happens is you bend the light, and your cornea does most of that. It bends the light so that a big giant thing can be reduced in size into a tiny little thing on your retina. Images, by the way, are inverted and reversed, so up is down and down is up and left is right and right is left. But it's okay because your brain knows that, and so it just interprets the information and everything is fine. They say that the brain learns to do that during the first week of life, which is why sometimes babies look confused about where sounds are coming from and so on. So 65%, roughly 2/3 done by the cornea. I've read estimates up to like 75%. I don't really know. Yeah, I'm dying right here. That's so fine. Hopefully I'll finish this annotated PowerPoint before I die. So most of it, whatever the percent is done by the cornea, and the rest is done by the lens. So light rays from distant objects are roughly parallel, and they have to bend just enough to go focus on the retina. Once you get under 20 feet, light rays are more divergent. All right, you can see in the diagram there. Therefore, they need more bending in order to focus on the retina. So you need extra refraction, which is the bending of light, and that's where the lens comes in. That's called accommodation, and it's done by the lens. All right. Suspensory ligaments change the shape of the lens to help focus the image on the retina. So for distant objects, the lens is thin and flat. In order to focus up close, the lens becomes more ball-shaped, as you can see in the lower right illustration there. That's what happens. When the suspensory ligaments pull on the lens, they take the pressure of the fluid in the eye off of it and allows it to bunch up and become thicker. So that's what's going on. Most of the time, if you're looking at things more than 20 feet away, your cornea does all the bending you need. It's once things get close, that's when you need more. And again, they say in newborn babies, brain may still be figuring out how to do that. That's why sometimes you get up close and you talk to a baby and they have that look on their face like, what the hell is going on? Where is that noise coming from? What is this thing? So, I don't know. That's just what they say. They say. So some common problems with refraction. Let's take a look here. Most refractive problems result from a misshapen eyeball. Blame your parents. It's the genes that does it. The emmetropic eye is the normal eye. And what happens here is it's basically a spherical eye. And when you look at something, the cornea will refract the light so that the image comes to a nice pinpoint right on your fovea centralis. Well, some people are myopic. They're nearsighted. And the way to remember this is when you describe it as nearsighted, it's what they see best. So people who have myopia see up close things really well. They can focus on things right in front of their damn nose. But when they're looking at things like, on the other side of the room, it's all blurry. They can't tell, wait, do I know you? Oh yeah, I recognize your voice. Your face is just a big blur. That's the most common visual defect. A lot of people wear glasses for myopia. Close objects are in focus, distant objects are blurry. In this case, what happens, you can see the illustration on the left, the eyeball is actually too long from front to back. It's kind of oval shaped. And so, as a result, the light focuses somewhere inside of the eye instead of onto the retina. So what you do is you put an extra lens in front of the eye, and that corrects the amount of bending that's done. It's so simple. People figured this out long time ago, centuries ago. Even ancient societies understood that, various kinds of things could bend light and help you see better. So you just use an artificial lens. You can either wear glasses where the lens is out in front of your face, or you can put the lens right on your eyeball. Of course, they call those contact lenses because they make contact with your cornea. Hyperopia is farsightedness. So remember, farsighted, that means that they see distant objects really well. Sometimes these are the people they call eagle eyes. These are the people that read that sign, like at the end of the block, and they can see all the words. You're going, damn, how can you do that? But they have problems looking at things that are up close. They can't focus on things that are close to their face. In this case, notice in the lower portion of the illustration on the left, their eyeball is actually too short from front to back, so it's kind of like squished in the opposite way. And once again, you correct with convex artificial lenses. So myopia, you need concave lenses. Hyperopia, you need convex lenses. All right, either one is possible. It's not a big deal. Usually, if we were in the classroom, I would take a show of hands, and a lot of people have corrected vision. Overwhelmingly, most people have myopia. There are maybe just one or two that have hyperopia. There are other vision defects as well. We are oversimplifying it here. These are just the simplest and most common. Many others occur as well. One of these other ones, by the way, is called astigmatism. This is where the corneal surface is not uniformly curved. So the cornea should be basically part of a sphere. It should be a nice, perfectly curved sphere. Well, what can happen is, as you see in the bottom left, an astigmatic cornea doesn't have uniform bending. It bends more in some places than in others. So what that means is some parts of the image are in focus and other parts are out of focus. Most people, by the way, who wear glasses, most people with myopia also have astigmatism. So they usually go together. It just means that, your glass lens, for example, isn't going to be exactly the same thickness. Some parts of it may be more or less thick than others in order to accommodate the non-uniform bending of your cornea. All right? And presbyopia, presby, by the way, means old, opia, eyes. So presbyopia are people who have old eyes. That's like Presbyterian, theory, Presbyterians. Those are the old theory people, all right? So that's one of the first Protestant religions, so Presbyterians. Presbyopia, the lens gets hard with age and cannot accommodate, so it can't bunch up. Remember, for close objects, the lens becomes kind of like a tennis ball. It has to bunch up and get thick. Well, as you get older, the lens becomes stiff. It can't accommodate, and therefore you can't focus on things up close. So it's the loss of ability to focus on close objects. Usually happens by the time people get into their 40s. They say, that term literally means old eyes. Easily corrected with bifocals or reading glasses. They say that basically what you do is you deal with presbyopia until you run out of arm length. So, you're at the point where when you're young, you can read things right in front of your face. As you get older, you have to hold them further and further away. You finally get to the point where even at arm's length, you can't read it, and that's when you either get bifocals, if you already wear glasses. So what they usually do is they make the bottom part of your glasses for up close and the top part for far away. Or you just go by, you go to the drugstore, wherever, Walgreens, and you can buy a pair of reading glasses. Like $10 now, I think. That's what I do. I buy reading glasses. I wear contact lenses. I have myopia. But bifocals are just, that's too fancy for me. I just go ahead and get the cheap reading glasses. I actually found some, they're like a pack of 3 for $12 or something. And they get bent up and they don't last long, but what the hell, $4 a pair. Mentioned by Aristotle in the 4th century BC, this isn't something people just realized. And glass lenses, again, used for presbyopia as early as the 13th century. So the idea of correcting vision has been around for a while. All right, so let's talk about where light goes. Penetrates the retina. All right, has to do that. Has to activate the photoreceptor cells, the rods and the cones. So rods and cones transduce light. Remember transduction. You have to take light energy, physical energy, and transduce it into nerve energy, into membrane potentials. So rods and cones excite bipolar cells. You can see there in the illustration on the left. There are lots of other kinds of cells in your retina. All right, so rods and cones excite the bipolar cells. Horizontal and amacrine cells integrate information from multiple photoreceptors and bipolar cells, and then they excite the ganglion cells as well. What's going on with all those other cells in there? Well, you know how, like, if you take a picture with your camera, with your phone, whatever, and put it onto your computer, and you can take the picture and you can adjust, like, the contrast and the brightness and the sharpness and stuff like that. That's essentially what all those cells in your eye are doing. They're basically fine-tuning the image before it ever gets sent to the brain. So think about that again, just in philosophical terms. You're not ever actually seeing the real world, so to speak. You're seeing a world that's been modified. The image has been modified by your retina before it ever even gets to the brain. So that's what's going on. Axons of the ganglion cells form the optic nerve, leaving the eyeball. No rods or cones at the spot where the optic nerve leaves the eye. That is the optic disc or the blind spot. You are technically blind there. Okay, optic disc or blind spot. So what happens once it leaves the eye? Well, they cross at the optic chiasm, and once again what's going to happen, the medial fibers cross, lateral fibers stay on the same side. That's kind of cool because it means that each eye sends information to both sides of the brain. So if you ever, some kind of accident, you lose vision in one eye, the other eye will still send info to both sides of your brain. Both sides of the brain get info from both eyes. Optic fiber synapse in the lateral geniculate nucleus of the thalamus, and then proceed to the superior colliculus for that visual tracking. That also then synapses with extrinsic muscles of the eye to control them, so you can follow a bird in flight with your eyes. And the optic radiations carry the signal to the occipital lobe where visual perception takes place. Your occipital lobe is where you actually figure out what it is you're looking at. You're making sense out of what you're seeing, all right? You're taking past information from your memory, you're taking current information, you're using all kinds of different neural resources to interpret what it is you're seeing. Optical lobe is where vision really happens. Now let's take a look at ultimately we're going to figure out about how you see colors. We have to start by noting that light is a type of electromagnetic energy, all right? It's an electromagnetic wave. X-rays, radio waves, TV waves, cell phone waves, ultraviolet light, infrared light, radar are also electromagnetic waves. Electromagnetic energy is one of the 4 types of energy in the universe or whatever. So, and they have a wavelength. Waves can be measured by the amplitude and by the wavelength, by the frequency as well. For light, they use wavelength. So notice you can see gamma rays down there, just a tiny fraction of a nanometer, all the way up to AM radio, which can be 100-meter wavelength. So again, wavelength is just the distance between the peaks of the wave. Well, visible light is actually a very tiny segment of the electromagnetic spectrum. You can see there, just a tiny little bit that they've then expanded to show you where different colors live. All right, so light just happens to be the particular set of wavelengths we can see, and other creatures can detect other wavelengths. So, for example, some animals can see ultraviolet light. Other animals can see infrared light. Like if aliens came to Earth, they might be able to see, radio waves. You would be sitting in the classroom, I would have the alien guest there, you would start texting on your phone, and they would be able to see these cell phone waves coming from your phone. We can't do that. We only see what's called visible light. But there's lots of other electromagnetic waves out there. We just can't detect them is all right? Now, it's not exactly correct, but you can roughly think of violet or purple light as being around a 400 nanometer wavelength. Green is around 500. Yellow is around 600. Red is around 700. Again, it's not exact, but it's just a rough guideline to kind of keep in your head so you know roughly what the different colors are, how they correspond to different wavelengths of light. So rods and cones are the photoreceptors. Photoreceptor, literally, they detect light. That's what they do. They detect light. Rods are shaped like rods. Who would have figured? You got about 120 million of them, they say, roughly. Rods can see shades of gray in dim light. They're good at detecting shapes and movements. So these are basically the photoreceptors that you use when there's not a lot of light available. And in that case, what's most critical? What's most critical is to see if things are moving, because that's, again, a survival kind of a thing. And the overall outlines and shapes of things. Is that a person? Is that a dog? What is that over there? So rods are not very precise, but you've got a lot of them, and they do see well in dim light. All right, only shades of gray. Rods can't detect color. Cones, on the other hand, shaped like little ice cream cones. All right, so there you see them. You only have like 6 to 8 million. Here's the deal. Rods are spread basically all over the periphery of your retina, but cones are really only in one place, and we already know where that is because I talked about it in the anatomy PowerPoint. Yeah, they're in the macula lutea, specifically in the fovea centralis. So this is where you get sharp color vision, all right? Sharp, crisp, clear color vision. That's why when you want to look at something, you turn your head and make that image fall directly in a place where the light bouncing off of it will hit your fovea centralis exactly. That's where you see best. The fovea of the macula lutea. Densely packed region of cones, all right? This is where the exact visual axis, that means when you turn your head to look right at something, that means the light bouncing off that image will fall exactly on your fovea centralis. And that's where you have sharpest resolution. That's called acuity. Visual acuity is the sharpness of your vision. Now remember we talked about dogs. Why did dogs have such better smell than we do? Because they had many, many more olfactory receptors. Well, what animal do you think of as having great vision? Well, a lot of people say eagles. They talk about having an eagle eye. What's going on with eagles? Eagles may have as many as 50 million cones. That's why they can hover high above the desert and see a little rodent down there running around. They have outstanding vision. Again, it's the matter of how many receptors do you have. The more receptors you have, the better your sense is. And that diagram on the bottom in the middle is good. It shows the distribution. Look at rods are all over most of your retina. Notice at the optic disc there's no rods or cones. You're blind there. And then notice as you get into the macula lutea, the number of rods starts to decrease and the number of cones starts to increase until finally you get right at that fovea centralis and it's nothing but cones. That's where you see best. So photopigments and rods and cones undergo structural changes when light hits them. So the basic unit of light, the photon, when a photon hits a receptor, they undergo a structural change. All photopigments involved in vision contain a glycoprotein called opsin and a derivative of vitamin A called retinol. That's why vitamin A is important for vision. Vitamin A deficiencies lead to night blindness, so that's called retinol. Retinol is the light-absorbing part of all visual photopigments. So, in other words, a photopigment equals opsin plus retinol. All right? Photopigment equals opsin plus retinol. So opsins are what determine the particular wavelengths that are absorbed. All right? Each opsin has its own special band of wavelength that it is most sensitive to. And we have 4 different opsins. So rods contain a single type of opsin called rhodopsin. All right? And again, that's for seeing in dim light. Really it can't really see individual colors, it just sees shades of gray, all right? Not the book or the movie, just the normal shades of gray. Cones have one of 3 different kinds of opsins. So any particular cone has only one of the 3. And each one permits only a relatively specific wavelength or color of light to be seen. So what's going on here then is we have 3 kinds of opsins, and therefore we have 3 kinds of cones because each cone has only one. And each cone specializes in a certain range of color. And let's take a look at that. There you go. There's the photopigment. There's the retinol and the opsin inside. Okay? So take a look here just at the absorption spectra. So look at where it says rod. Okay? That's rhodopsin. All right? So that's rods can see over a wide range of wavelengths. So that's why they're good for night, because they see a wide spectrum of light. They're also very sensitive. L-opsin is most sensitive to red wavelengths. So L-opsin, therefore we have L cones, which are L wavelength. And why is it L? Because long. Remember, as I said before, wavelengths correspond to colors. Red is up around 700 nanometers. That's the longest wavelength. So that's why it's called L-opsin, L for long. L-opsins are really good at seeing red, all right? Then M-opsin is really good at seeing green, all right? Notice somewhere there in the 500 range or thereabouts. And the S-opsin, S for short wavelength, is best for blue. It responds best down there around 400 nanometers. So again, rhodopsin found in the rods, sensitive across the entire spectrum. But what you need to do then is when there's a lot of light available, you're able to use your cones. Cones don't work in dim light. Rods do. If there's sufficient light available, all right, then you will have your cones responding, and each one responds to its own special narrow band of wavelength. And you're going to put all three of those together to get information about color. So as many as 100 rods may converge on a single ganglion. That's why they don't have good crisp vision. Whereas each cone stimulates only a single ganglion cell. So there you go, acuity. Rods do not have great acuity. Cones have fantastic acuity. Combining information from all three types of cones produces clear, crisp color vision. Now, you may have heard of people who they use the word colorblind. What does colorblind mean? What does that usually mean? Does that mean they can't see any color? No, hardly ever. It's very rare to have the condition in which you can't see any colors, when you can only see shades of gray, not the movie or the book. Normally what happens is one of your cones is not working. So a typical colorblind person, they were born with only two of the three kinds of cones. Therefore, they're missing part of the visual spectrum. Therefore, they still see colors. They see an impaired range of colors. All right? So let's take a look right now. Here are some standard color blindness tests. The Ishihara is one of the standard ones. It used to be anyway. I don't know anymore. But they have these various color plates. So look in the upper left. Hopefully you see an 8. If you don't, you might see a 3, or you might see nothing at all. So this is one of the ways that they can tell which of your options is not there. If you have all three options, you'll see an 8. If you're missing one of the particular options, you'll only see a 3. If you're missing another one of the options, you won't see any number at all. Isn't that clever? So individuals who have all three types of cones are known as trichromats. Tri, because we have all three cones. They have full color vision. Colorblind are missing one or more types of option. All right? As a result, they see some colors, but not the full range. Most are dichromats. So they have two, they're just missing one. Now, red-green color blindness is the most common type of color blindness. This is a sex-linked X chromosome. So what does that mean? Remember, if you're female, you're XX. If you're male, you're XY. So females get 2 chances to have good color vision. Males only get 1. So if you're a male and you got a bad X chromosome, you're going to be colorblind. And by the way, who gave you that X chromosome? Yeah, you got your Y from dad and your X from mom. So if you're colorblind, make sure on Mother's Day, send your mom a card. Hey mom, thanks for the color blindness. No, she's your mom. She loves you. Be nice to her. So red-green is a defect in either the L or the M. Now why either L or M? Well, notice it's called red-green color blindness. If you take it in art class, you may know about opposing colors, about color theory. All right. So, you know, the color wheel, the color thing. So, you know, what's the complementary color of red? Green. So what goes on here is if you have problems with either red or green option, you have problems with the complementary color, all right? So people with red-green color blindness have difficulty telling the difference between reds and greens. And I've seen this in person. I had a guy in my class a few years back who had a red-green color blindness. And normally, you guys in virtual don't know, but when we were in the classroom, I had models, and I would mark the models with different colored pins, you know, and I had a blue, a green, a yellow, a black, a red, whatever colors I didn't say, orange. And then I would have a little, like 3x5 cards, say, you know, name the item with the red pin, name the item with the blue pin, and so on. And I had to remember for him not to use a red and a green pin on the same model. And I would forget occasionally, and he would call me over and say, Tom, you did it again. He could not tell which one was the red pin and which was the green pin. That's typical red-green color blindness. Difficulty distinguishing reds and greens. Blue-yellow is fairly rare. It occurs, it's not sex-linked, all right? It's a defect in the S cone. It occurs on chromosome 20, I think. I don't remember. I knew that at one time. These people have trouble telling blues and yellows. Again, this is relatively rare. It's equally common in men and women. But red-green color blindness, because it's on the X chromosome, this is mostly men. And I forget what the statistics are, and they keep changing. Something like 1 out of 10 males is colorblind. That's why whenever you want color advice, always go to a female Everybody knows that. They see colors much better. Males are terrible with colors. But I've been watching Project Runway, and I'm getting much better. I'm learning more color names like seafoam and aquamarine and puce and fuchsia. I'm working on it. So notice there in the upper right of the illustration, there you see a 7. All right. So hopefully you can see that. If you can't, you may have a red-green color blindness. In the lower left, what's going on there? 35. So as it turns out, back in the 1970s, we were at war in Vietnam, and we had conscription back then. We had a draft. You had to go. You didn't have a choice. You had to join the army. Well, a lot of people didn't want to. They said, I don't know anybody in Vietnam, and I don't wish any of them harm. So people, when they learned that you couldn't be colorblind, they would try, when they took the color blindness test, they would try to fake color blindness. And so what they had to do is they had to put in a distractor item, that number 35. Turns out, no matter what kind of color blindness you have, you can see the 35. So they would show that plate to people. They would show, like the top 2 plates, the one with 8. What do you see? I don't see anything. All right, what number do you see there on the top right? No, I don't see anything. What number do you see in the lower left? I don't see anything. Ah, busted. Grab your rifle, son. You're going to Vietnam. So, and then what about the bottom right? That's where you can only see it if you're colorblind. In other words, seeing colors actually distracts you from the shape of the number. I think it's at 27, something like that. If you kind of look, you can kind of tell. But yeah, so people with certain red-green color blindness can see that number. They can't see the other ones, all right? So individuals see a 3 or an 8, depending on which cone is defective. Distractor, people are faking color blindness. Numbers visible only to individuals with a particular defective cone. And there you go. That's don't use that in your color blindness test. You may upset people.